Well, welcome everybody to the next episode of the Good Medicine Podcast. I'm very excited having today one of my really good friends, Dr. Greg Critchfield. So Greg is a pioneer in the field of genetic testing and molecular diagnostics. And this is something different than we've talked about previously. I think a lot of attention gets put on the therapeutic side of medicine, and a lot of interest on that side. But really, half of medicine is just trying to figure out what's wrong with people. The diagnostic side, it's maybe more than half of medicine is that. So that's really where Greg comes from and his background. And he's had some amazing experiences in his career. He was early one of the executives at Myriad Genetics, which was one of the pioneers in genetic testing. And so we can talk a little bit about what genetic testing is and how you develop those tests and what they turned into. He actually did something was involved in legal battles that went all the way to the Supreme Court, which is pretty amazing. Not many people can say that, and I don't know if that's a good or a bad thing, a badge of honor or not. But some of the work that Greg has done has moved all the way, has really moved the whole field and really set the precedents for how a lot of this stuff is done. So we're so pleased to have you here, Greg. Excited to have a good conversation with you today.
Dave, it's great to be here, I'm excited to share a bit about my experience, and I hope that people can find some thoughts and some inspirations about ideas that they want to pursue. Hopefully that will come out of our discussion.
Absolutely. And I think that's exactly what we're here for is to give people some insight into things that maybe they don't think about all the time. And we start out, I think, in our careers with a certain thing in mind, and we usually end up in a different place. And I'd love to hear how you actually started out. I know you're a physician by training. You have an MD. And how did you end up wanting to develop diagnostic tests?
That idea was planted in my mind when I was a teenager.
Oh, wow.
My father was a family practitioner.
Okay.
And I remember sitting down with him one day and saying, "What's the hardest thing that you do?" And he goes, the therapies, they're prescribed, you do them, you use them, you know what to do. He said, the hardest thing is figuring out what the patient has.
Yep.
It's the diagnosis.
Yep.
And I decided that I would want to make that a focus later on in my life. I had a number of really interesting experiences. I became very, very fascinated with the advent of technology, computers. I did programming at the University of Utah. I took a class when I was a junior in high school. I knew that I'd do something in that area, and I had a chance in my career to pursue that track. When I went to medical school, my advisor in medical school was Homer Warner.
Of course, yeah.
And so
incredible.
Homer was an amazing inspiration to me. I had a chance to walk through his laboratories. I was walking through one day, and the computers they were using at that time were mini computers by Data General. That's what they were. And these little Data General computers would be slaving away at things, trying to compute them. And I remember how slow they were. And I said, "Why is this running so slow?" And he said, "Well, it's running a program by a geneticist that we just recruited here, a guy named Mark Skolnick."
Oh, Mark Skolnick.
And Mark was doing linkage studies and trying to figure out locations of genes. He was a gene hunter.
Yep.
And I thought, oh, that's interesting. I put that away for a while.
Yep.
And then when I went to medical school, I remember talking. You've chosen a very good scientific track for yourself. I remember thinking after the first 2 years of medical school, the basic science years, I thought, is that really all we know? I was disappointed that we didn't know more.
Yeah.
And it became clear to me that we needed to push the envelope and do new things. And so when it came time to actually interview for residencies, I decided that I would go somewhere where I could get training in pathology, because pathologists were sort of the physician's scientists.
Yep.
And in fact, one of the lines I used when I was interviewing was really funny. They said, "Well, why do you want pathology?" I said, "Well, because it's the closest thing to science and medicine." and it was true at that time.
True, yeah. It still is true.
It's still true. And so my journey took me through a series of things. I actually ended up, first year was at University of Michigan doing general pathology. The next 4 years were at University of Minnesota. They had a very strong focus on laboratory medicine using data. And in fact, I did a fellowship there with National Library of Medicine in computer science. I studied computer science, mathematical modeling, and statistics. Those are the three things I spent my time building my graduate program. And that became the basis of everything later on. I had a chance after that to do an academic job at Wayne State University, and I actually cajoled the chairman into buying a computer for me, so I could actually program things. It was a Sun Workstation in those days.
Sure,
you remember Sun?
I remember Sun Workstations, absolutely.
And he did, and I was there, but I had written a paper that somebody reviewed, a guy named David Eddy. David Eddy was the world's foremost mathematical modeler in health policy.
Okay.
He was in 1980, 81, he actually put forward an idea. He did an analysis and found that if you actually changed the frequency of pap smears and used part of the money you normally devote to pap smears to educating women, it changed the American Cancer Society recommendations for screening.
Interesting.
He was given an endowed chair at Duke University, and he read my paper as a reviewer after he turned the review, and he called me up. He goes, "Look, I'm doing something similar to you. You need to come down here and look at a job down here." And so I said, "Well, I've got things lined up. No, I'm not going to do it." And then he called me a year later, when I was at Wayne State, and he said, "Come down and look." And I went down, and it was fascinating what he was doing. He's building Bayesian models to assess the state of evidence so that you could prove to a payer it was time to actually cover it. Way ahead of its time.
That's way ahead of its time. That's what's today, right?
It's here now.
Yeah.
And those principles were ingrained in me. I joined the center as the associate director for medical affairs there, and I worked with David. And we were building models for insurance companies, and those models became the basis of how I began thinking about laboratory tests and novel technologies. Mm-hmm. And eventually it led to some principles that I was able to apply later. Where if you have a new technology and you apply it, somebody's going to pay for it. So the value has to be reflected in what they pay.
Yep.
And it has to be able to improve the well-being of human beings. And what are all the consequences economically and clinically when you do it? That means you have to be able to design the right studies to answer those questions or get the right evidence so that it's meaningful. So that became really a core strategy that I used after that.
Well, incredible.
I practiced medicine after that. We were going to build an academically based business and publish papers doing work for payers and things. But the funding was hard in those days for this kind of thing. And we decided it's time to do something else. So I came back and practiced laboratory medicine pathology at Utah Valley Regional Medical Center.
Oh, is that right? I didn't know that.
Yeah, I was there for almost 9 years.
Wow. Okay.
And then I was recruited to Quest Diagnostics, where I went in and was replacing the former chief medical officer Ray Gambino. I became the chief medical and science officer of Quest in it was in 95 through 98 that I worked there, and we were developing new technologies. But as I did this, it became clear to me that Quest was a great place to work. I learned a lot on the business side. I learned a lot about technologies, but frankly, the technologies were already final form technologies. Yeah. They'd been developed by other people.
That's right. Yeah.
And I decided it's time to go earlier, and that's when I went to Myriad. And joined Myriad. So that's how that part of my story.
So how did you reconnect back with
Mark?
With Mark, yeah.
It was 1990. Let's see, it was 1997. I was flying into Salt Lake just to visit relatives. And I decided to. Somebody said, "Well, why don't you go talk to Mark Skolnick and Pete Meldrum?" And so I sat down with them, and I gave them my view of the world, how laboratory tests inform medical decisions. Exactly what you're saying.
Yep.
In fact, some data that came from Intermountain showed, and Institute of Medicine, other people who quote this statistic all the time, 70% of the data that you have is laboratory based, either imaging or molecular, and that's how people make decisions. And so I gave this little speech to Pete and Mark and talked to them about it. And then later on, a few months later, they said, we have a position that we're interviewing for. Would you consider coming in and being the head of the diagnostic company? And I came out and looked at it. It made sense to me. And I decided, let's do it. And that's when I joined Myriad in summer of 98.
Oh, that's incredible.
Yeah.
So summer of '98, you think about that. That's Human Genome Project's in really ramping up, and there's a big thought about how's this going to change the world, right? We right now we don't know very many genes, and in a few years we're going to know all of them. And how's that going to change everything? And I'm sure it was a pretty exciting time thinking about. Like you said, Mark spent his whole career searching for genes, and now we're going to sequence all of them. We're going to know all of them. So it's pretty exciting times right about that time. And what were you thinking? The first thing that the application of genetic testing and how that was going to get incorporated, because it's still pretty darn new right at the time.
It is new. Yeah. And I was thinking about this as I was driving down today. It's interesting the amount of fear that people had of genetics.
Yes. Yeah.
It was an unknown field. They didn't know if people would become despondent if they found out they had a gene, if they'd inherited a mutation that could cause problems. We didn't know anything in those days. Yeah. And genetic counseling was in its infancy. Yes. We'd had work with it in looking at fetal anomalies and counseling parents that had children that were born with inherited disorders. Yes. So there was that basis, but it had never been done in cancer genetics. Yeah. And so one of the things that I decided when I got there is we had to build something that do the right thing for patients, the right thing for doctors, and we had to get things ready. So one anecdote that you'll like. John Nelson was a very prominent physician in Salt Lake City. He was president of the AMA at one time. And I talked to John and I said, why don't you bring your guys out from the AMA and let's talk about genetics because it's going to change the world. And he brought people out. Yeah. So he brought his head of research, his head of education, a couple of the officers and himself. And I gave him a tour of the lab and said, this is what we're doing. And I said, he said, well, what's your plan to tell doctors and patients about it? I said, well, we're going to spend the first part of our tenure talking to doctors so doctors understand what this is. Yes. And then eventually, at the right time, we will be going out and telling consumers about it because they need to know as well. And they almost went as white as a ghost because they're so worried about what this would do. Yeah. And I said, but you have to understand that there will come a time where if people don't adopt new technologies, they're at risk. And in medical school, we learned don't be the first to adopt and don't be the last. That's right. And that's really the mindset of many, many people. Yeah. And so you have to have the right information to the right people. And we spent a lot of time developing policies, procedures that would protect patients, protect confidentiality, that would educate doctors, so that, and it would plug people into systems that could help them to actually absorb this information and use it. Yeah. So that's what we had to do.
That's incredible. Yeah. So you think about the real explosion for Myriad happened with the breast cancer susceptibility discoveries, and so the Utah, if people don't know, Utah has been really one of the leaders in the world of genetic discoveries. And we have a unique resource here called the Utah Population Database, which allows us to identify families that have specific diseases running through the families and allow us to be able to try to find, the genetics associated with that. So that's, the old school way of doing things is you find the family and you try to trace back what's moving through the family. And so some work was done, foundational work here in Utah, that discovered two genes that were responsible for hereditary breast cancer syndromes. And they have become, in our world, they're household names. But I think even generally, most people know about breast cancer susceptibility, and that's due in large part to the work that Myriad really did. Yeah. And so tell us about the discovery of the BRCA1 and 2 genes and how you translated that into a genetic test that was available to go out and to screen individuals for their risk for breast cancer.
Well, one of the interesting things in those days is that we didn't have a deep understanding of segments of the genome to be able to do a lot with it. People had found biomarkers here and there. And one of the big debates that took place at Myriad, this is before I got there, but it was actually a very fundamental debate, and that is, do we do a sequencing test where we sequence everything that we know, or do we just do point mutations and build a panel and use microarrays? That was one of the initial debates. The company, thankfully, had invested in the laboratory, invested in the technologies. They took the gene discovery technologies and turned it into a laboratory process that was bulletproof. It was correct. and ultimately scaled it up. And I can give you some scale up stories in a minute. but the idea of sequencing was really important because and what it means, if you had no information and you started sequencing the gene today with no information at all, 40% of the time you would find variants of unknown clinical significance.
Yeah.
And you would be paralyzed by that.
Yes.
Because you'd say, well, that's almost 50% of the patients you would screen would have a variant where you say, you got something that people don't have, but we don't know what it means.
We don't know what it does. Yeah, that's right.
So we spent, one of the things that my team did, we built a database. We worked really hard to characterize mutations. So we made the decision that we would track a patient and revise reports throughout the entire lifetime of that patient. And we did everything we could to keep them in touch with them. And if their new variants were discovered and then they were characterized, we would actually tell them.
You inform them.
Yeah. We'd inform them.
Yep.
So we built a living, growing compendium of gene mutations and watched it over time and built it so that we could identify and characterize genes. So the 40% got down into the low single digits of variants with the work that we did. That database became incredibly important. We were able to classify things that we saw that nobody else had. There were also some cases, and I won't go into specifics. I'll just give you generalities here. There were cases where well-known centers had actually found some mutations or called mutations, and patients had irreversible surgeries, and when in fact they were not deleterious mutations.
Ouch.
and there were lawsuits because of that, of course.
Yeah, I can imagine.
And so you had to be really careful. Everybody was waiting to see if Myriad, because we held the gene patents, the spotlight was on us. People were waiting to watch to see if we would stumble.
Yeah.
We were so careful. We had to do everything perfectly. And as we told everybody in the lab, this tube of blood that comes in, that's a person.
Yeah.
Do it right.
Do it right. That's right.
Pretend it's your spouse.
Yep.
It's your girlfriend. It's your friend. Do not make mistakes because this has impact on this person. You know.
Incredible.
Yeah.
So walk us through kind of the launch of this.
The test was actually launched on a research basis back in 1990, 97, 96, end of 96.
Yeah.
October of 96, right?
Okay.
That's what it was.
Yeah.
And I joined Myriad in the summer of 98. At that time, the revenues were de minimis.
I can imagine.
It was just we were doing research, and there were several interesting things that happened. We were, a lot of people were watching what was Myriad going to do with these patents. Well, we didn't really patent the genes. We had patents on the process of actually cloning the gene, and it was a derivative of the gene that we'd actually patented stuff on. So that's something that people don't understand. And even the Supreme Court decision decided to ignore that. You know? Okay,
so you can't patent gene. Well, we didn't patent.
We actually didn't. You know, so that's kind of interesting.
Yeah, that is interesting.
But it made for good sound bites, and people could say things about it.
Yeah.
But what we did, we worked hard as we first went out to gain support of the research community. And there was a cancer genetic network. There were 12 centers in the U.S. that were doing work with genetics, and they would collaborate on things. Some of them were involved in the early research with us as collaborators. Most were not. Okay, but what happened is there was one center that decided to offer its own laboratory, its own laboratory test. It wasn't as good as ours. Yeah, it wasn't as comprehensive. And it was expensive, and they were charging the other centers. And we went to Richard Klausner, who was the head of the NCI at the time, and said, "Richard, that's not right." He agreed with us, and so we did a memorandum of understanding where we actually made our test available to anybody that was NIH funded for any grant at all. If they wanted to get sequencing, we'd give them a highly discounted research rate, and then we would not patent anything that we found. We'd let them publish and do what they wanted with it. That led over the ensuing 3.5 years or so after the memorandum of understanding was signed. That led to 11,718 sequencing tests being done.
Wow!
And those tests were available to the scientific community. We built a mountain of information. Yeah. And that became the basis of numerous studies that showed what knowing early the difference it makes. And it actually helped to propel the field forward because we had generated more scientific data on the clinical utility.
So how did your investors feel about that strategy?
Well, this is a great story. One of Pete Meldrum's initial ideas with Myriad, and Pete was a very good and solid leader. His idea was a threefold strategy when Myriad went public in 1996. Okay, number one, discover genes that are important. Number two, develop diagnostic tests and generate revenue, and with those revenues, become a fully integrated pharmaceutical company.
Yep,
that was a threefold vision of Myriad, and that's the vision it had when I joined the company.
Okay,
and so the investors, we had a split base of investors. Some were interested in therapeutics, some were interested in diagnostics. And so it was really interesting to watch what happened over time. And as our diagnostic revenues grew, and by the way, my team grew the revenues 54% compounded annually for 12 years in a row.
Oh my gosh, that's incredible.
It was amazing.
That's amazing.
They did a great job.
Yeah.
They did a great job. And I remember going to one of the investors that had invested because of the pharma play.
Yeah.
And sitting down with him, and he looked at me one day. He says, your revenues are actually growing a lot, aren't they? And I said, I've been telling you this for 3 years.
You're just now noticing.
You just noticed it. Exactly. So I think that the investors were two-minded, depending on which one they liked. But it became clear eventually, with the growth that happened, the tremendous growth in genetic testing that occurred, and the paradigms that we built, it grew pretty fast over time. Yeah.
So how did you get reimbursement? That's always the hard part about diagnostics, right? Is that that it's getting it approved is hard.
Yep.
The FDA has to say, yeah, this is good, this is reproducible, but getting somebody to pay for it is maybe even harder than that.
Yes. That comes back to the work that I did with David Eddy. Okay. And what I had a back of the envelope calculation. Back in the early days, it was the lifetime treatment cost for one case of breast cancer. At that time, were $150,000.
Yeah,
and so the argument you made to a payer was, these patients are already in your premiums. You've already paid for them. If we have a test that can identify them earlier, and they could be diagnosed at an earlier stage, or even eliminate the risk of breast cancer, isn't that more valuable to you? That was the back of the envelope calculation. That model is what actually began selling it. And it was interesting, in the early days, the first major payer that came in was Aetna.
Okay.
Arnie Cohen was the head of women's health at Aetna. And Arnie looked at this and he said, we've decided that we would pay for a genetic test on an individual who is not an Aetna member if their family member in Aetna had a documented mutation that is autosomal dominant for BRCA.
Wow.
We would pay for testing that other person.
That's incredible.
Isn't that amazing?
That's amazing.
Yeah. And so if the I'll restate. I want to be sure I got it right. I'm going to restate it. The policy was that if there's a patient that you want to get a genetic test on, if they have an individual outside the family with a mutation, they would pay for it for sure. And so it was very expansive in a way, and he decided that would be something they would do. They announced it. We got an educational program together with them and the AMA together. We actually talked to people about what it meant, and that was a big part of our launch, actually.
That's incredible.
And there were other payers followed. Kaiser followed suit. They did some work and published it. We had all the major payers came on. United came on. Everybody came on eventually, and once they came, it became clear to the smaller payers that didn't have the sophistication and the resources to really dive into these things. It became clear that this was the way it needed to go.
Yeah.
But the biggest fear in those days, in the early days, was the fear of not knowing what to do, and the fear of genetic discrimination.
Yes,
those were the two things.
Yeah.
And there were genetic discrimination. As it turns out, it did not occur to any great extent at all, even though there was a fear of it. Eventually, there was legislation passed that said you can't discriminate against somebody on the basis of genetic changes. So that helped. But by then our revenues were huge anyway. So we were growing by that time.
Yeah. Well, that's amazing. So it still is something that people worry about, though, right? That the fear that if I find out I have some sort of risk that's going to be held against me somehow. So
let me address that though, because there's a therapeutic angle to this, as you know.
Yeah, absolutely.
Okay.
Yeah.
One of the things that I did, I talked to Pete back in 2006. I said, look, we need to be talking to people that are developing PARP inhibitors. And for the audience that doesn't know, PARP inhibitors they inhibit some of the processes that are involved in DNA repair. They allow, if you actually inhibit the PARP genes, small nicks can occur, and eventually they'll get enough of them that you'll get a break in the strand, and it's a problem. Because the way that genes are inherited, autosomal dominant inheritance of BRCA genes means that there's a defective copy if there's a bad mutation, and there's a normal copy. The normal copy is intact, but if that cell is hit by a damage, it can lose that, and that's one of the reasons that cancer can develop more readily in somebody that has a hereditary mutation. It's because you get a gene from the mother and the father.
That's right. So it goes from having to lose two things to just having to lose one thing. That's right. BioHive is a nonprofit building the future of healthcare innovation right here in Utah. As one of the nation's fastest-growing life science industries, they bring together scientists, entrepreneurs, students, and companies to create breakthroughs that save lives and strengthen our economy. Whether you're in a lab or at a startup, BioHive is here to build, brand, and bring together a thriving life science community, and there's a place for you. Learn more, get involved, and see their latest happenings at biohive.com.
That's the two-hit hypothesis that happens. You have that problem, then you get hit by something outside that damages the other gene, and the two-hit enables the cancer to occur. So some smart researchers had the idea. Well, look, if we can actually inhibit the PARP, if we can do a PARP inhibitor on a tumor that has one defective gene and one normal, and if we inhibit it, maybe those patients will respond. And as it turns out, the patients with ovarian cancer and breast cancer are, in fact, responsive to PARP inhibitors. And it's exploiting the very discovery that was made of what these genes did.
Yeah, so it's remarkable. You can actually turn it into a positive to say you're going to be uniquely sensitive to a treatment based off of your genetics. So instead of thinking it's a bad thing universally to say I have this risk,
it could even be better.
It could be better. That's right. Exactly.
Right. That's right. Yeah. And as it turns out, that was the first companion diagnostic use of BRCA testing. It was the approval that we got with AstraZeneca.
Yeah, with their PARP inhibitors. That's incredible.
It's kind of interesting.
Yeah, it's really interesting. So I guess taking just a step back and looking, so you put yourself in, the early 2000s or in 1999 when things are really just kind of taking off. Are you disappointed how far we've come? Are you surprised that things haven't taken off the way that you expected, or you think that that we're better than you expected based off of genetic testing being accepted and part of the diagnostic tool set that people use for diagnosing medical conditions?
We're far further along with regard to what we do with the breast cancer genes than other genes.
I totally agree with that.
So I'm glad that we've come this far with them. Is it universal? No. Does every patient know about this, and can they take advantage of it? Well, if they dig, they could, but they're not always aware of it.
Yeah.
And a lot of this has to do with the way our healthcare system works.
Yes.
So, how data are stored, how people are reminded, how we take care of people,
and the incentives to do it.
And
right?
incentives. Yeah. And you run into particular problems with genetic mutations like this, the inherited ones. Family members may feel ashamed and may not want their other family members to even know about some of it.
Yeah.
And that's part of the problem. A lot of this is education.
Yep.
And so I'd say we still have a fair amount of distance to go on the education part.
And I think, part of what, we're working on right now is a genetic risk for Alzheimer's disease.
Yes.
It's well understood, well studied. We know what the risk looks like, but getting people to actually run a test and understand that is challenging. And especially in the United States, it's really not taken off dramatically. And part of it is people say, well, if there's not a therapy associated, I don't want to know, right? Why would I want to know if it's not going to change?
Yeah.
So if I go to my doctor and say, hey, I ran this test and this is what it says, and your doctor says, well, I don't know what to do with that. I mean, I get that, but I guess you and I are probably of the same opinion that I want to know everything that I can know, because even if there's not a specific prescription that a doctor can write, there still informs you that this is me. This is my genetics, and this is what and it's not fate, right? Not everybody. In fact, it's quite interesting. Not everybody that has BRCA1 or 2 mutation actually gets breast cancer.
That's right.
And so you're not fated, but it does tell you have to think about this, and you have to, it's something that you have to pay attention to.
There were huge debates about genetics, and even a national panel that was convened to talk about a lot of these issues. And one of the things that comes out of this is that as you pointed out, there is information there. You may make lifestyle changes based on something. You may choose to do one career versus another. There are lots of decisions you can make that go beyond having a specific therapeutic that can actually nail the condition.
Yep.
And by the way, as I showed before, sometimes these discoveries will lead to things that can actually address that mutation and do something with it.
Exactly.
So if we don't look, we won't know.
Yep.
And that's part of the problem.
That is part of the problem. I think that's exactly right. And, so there are countries in the world right now that are doing genomic sequencing of their entire populations. And I would have assumed that we would have been further along with that, because we've had the technology to do it. It's gotten a lot better, and it's gotten a lot cheaper, which I think is what's driving more of it, because the economics really, back in the day when, we're talking about sequencing a whole genome back in the late 90s was almost impossible. it was very, very challenging. It was very expensive.
Yep.
And today, if you want to, there's places online you can send in a sample and get I have a machine downstairs in my lab. I can sequence you tomorrow, right? I can sequence your whole genome. So it's, we've advanced a lot in technology. And I'm excited to see what this does in terms of helping us understand how all these risks work. And one of the things I'm very excited about is in the past, we've known about genetic risk factors for all kinds of diseases, and we've done a lot in cancer, and we know a lot about cancer. We know I could probably rattle off at least 25 known risk factors that we know if you have this particular gene variant, your risk for cancer goes up. But what we don't know is why some people remain resilient in the face of bad genetics. So not everybody that inherits as a bad copy of a gene actually gets the disease. And understanding the whole genome actually helps us, because we inherit a mixture of things from our moms and our mothers and fathers, and that mixture can give us susceptibility, give a protection. And we know we've studied a lot about susceptibility. We've not studied that much about protection.
Well, yeah, and now you're getting into the other area that really fascinates me, and I spent time in my career on too. The genetics revolution has been enormous.
Yes.
And genes are like a computer program. It tells you, but it tells you what could happen,
That's right.
not what will
what's
happen.
going to happen. That's right.
That's right. Now you can go to proteins. And proteins are what the genes are actually designed to effect, and the proteins are really the worker bees.
Yeah,
they carry out the functions that are dictated by the proteins. So you go from gene to protein, and then you can, if you look at the proteomics, and can actually assess the proteomic expression. That gets you further along.
That's exactly right.
And at Diagnostics, that's precisely what we did.
Yeah.
We took that approach. We decided to look at proteomics, not at genetics, because the genetics of prematurity, the biggest problem in pregnancy, they didn't shed much light on what was going on. And so, but proteomics can. And today, the third part of that revolution is epigenomics.
Yep.
As you know,
yep.
Methylation and changes and all these things are part of the story. And even beyond having anti-susceptibility genes, protective genes, and other things, all this stuff comes together in a very complex way. We don't fully understand all these elements yet.
Yeah.
Someday we will, but we're not quite there yet.
And what do you? And then this is a great segue, because I was going to ask you. We call this multi-omics, right? We got genomics, proteomics, epigenomics. We put all those things together. Microbiome. There's all kinds of stuff out there. We put all that information together, and we call that a multi-omics approach.
Right.
But the complexity of that is so daunting. I mean,
yes.
You and I looking at that on a spreadsheet, we'd lose our minds. We couldn't figure out what was what. But there's a revolution happening right now with artificial intelligence, where AI loves this kind of stuff. You're feeding it patterns and saying, here's this. This person got sick, this person didn't get sick. Tell us what's happening right now in the world of trying to apply that data analysis to these very complex data sets.
Yeah, we even use some of that in doing discovery work in the areas that I've mentioned. I think that we're going to see more of it. I don't know if the predictions are going to be precise enough to make sense out of them yet. I just don't know.
No, I don't either.
The hope is that if we knew everything, we could predict everything. I don't know. I don't think that's true yet.
Yeah. I think it still goes back to an earlier comment that you made. It is disappointing how little we actually still today know about biology. Right?
We know a lot more.
We know a lot more than we used to, but there's still so much that we're just scratching the surface.
Well, and you look at it in drug discovery that you're doing. You're coming up with you're addressing mechanisms in ways that have not been done before with new molecular entities to try and change the way that things interact. Very hard to do. And as we know, very risky. But in fact, the benefits to the patients can be enormous if you hit the right things.
Exactly. It's funny because we think about with how little we know and how risky it is. It's a miracle we actually figure out what we figured out. I mean, it's remarkable. But I tell people the thing that I love about what I do is if I discover something, it literally can change the world.
Yes,
like drugs change the world. It changes the lifespan of people. It changes families. I mean, the reason why I'm involved in this is because I have a family history of colon cancer. I don't have any of the known risks for colon cancer, but I have multiple people in my family who have died of colon cancer, and so that worries me, right? And I don't want other people to lose their moms and dads and grandpas, and so that motivates me, right?
I have a colon cancer story for you from Myriad.
Sure,
that you'll like.
Yeah.
Okay. After launching the BRCA genes back in '96 and seeing those move along, we decided to launch testing for the Lynch syndrome genes, the mismatch repair genes. And for the audience, these are genes that if you have mutations in them, your risk of hereditary colon cancer, hereditary endometrial cancer, hereditary ovarian cancer is much higher than the general population. Those mutations occur with a frequency that approaches the BRCA genes. Not quite as much, but they're still very significant. And in the early days, what people did, it was actually an epidemiologic model for screening the population. You'd wait until somebody got cancer, then you would test them. If they had the mutation, you'd try and test their family members. Now, and that means a lot of follow-up with the genetic counselors and everything else. The very best program that existed was in Columbus, Ohio, and they would get about 1.2 family members per index case is all they'd ever get to do the test. And so I remember we had a panel of experts. They were gastroenterologists, people that were involved in cancer genetics of colorectal cancer. And we convened a group of them. And I remember sitting down with them. I said, okay, how long do you think it's going to take for us to sequence the majority of people in the U.S. that are carrying these mutations in their families? And they said, oh, it's going to take about 10 years. And I said, no, not really. If you actually run the math with the best follow-up that exists published, I said it's going to take 1,250 years.
Oh my God.
This is shameful.
It is shameful.
They were shocked, but they realized that we had to go to a different model. And so what we ended up doing is actually building a health economic model. I actually touched base with David Eddy and his group in California at that time, and they did an analysis, and they showed that if the probability in a person based on family and personal history was above 5%, you're better off going straight to gene sequencing. That became the standard, and the American Gastroenterological Association adopted that standard when the day that paper was published. That's how you change the world.
That's how you change it. Exactly right. Yeah. So I think that it's interesting that like I said at the outset, I think that the therapeutic side gets it's a lot flashier. Just think, I said drugs change the world, but diagnostics can change the world as well. And you think about just most of the experience that an individual has with a doctor is you go in, you get some blood drawn, and you don't really think about what's happening with those samples and what's going on. But there's a lot of sophisticated stuff that is being run on just a simple blood draw nowadays that can change the course of that physician gets that information back. There's things that they're going to do that can change your life individually and change the world. So it's equally as exciting, I think, even though, like I said, it maybe doesn't get as much airplay in terms of how people look at it, but the diagnostic side is, I think, we have is equal or even greater opportunity to change things by developing better and more sophisticated ways of analyzing what's wrong with people and diagnosing what's wrong.
Exactly. The better we understand the physics and the biology, the more we can benefit the patients.
Yep.
That's the way it is. I've got another example. There's a company I'm working with right now from Utah. Came out of the University of Utah. They have a way of diagnosing a condition that affects as many as 60 million people in the U.S.
Wow,
it's called laryngopharyngeal reflux. Everybody knows about heartburn reflux.
Yep.
But there's a form of reflux that goes all the way up into the upper esophagus. Mm-hmm. There's no easy, non-invasive way to actually detect it. And this company, out of a graduate student program, developed a way to put ultrasound sensors on the neck and just shoot ultrasound waves across and actually document that there's a bolus in the upper esophagus non-invasively. Never been possible before.
Interesting.
That's another example. And if you look at these things, if you can sort people into proper categories, those that actually have the condition versus those that don't, instead of wasting time on empirical treatment with no diagnosis, failures, patients getting frustrated, having to visit doctors as many as 10 times in a year and not getting anywhere, you can get the diagnosis nailed down. You can give them proper targeted therapy and not waste time and money and get the other people worked up for the other conditions that they have. That it goes back to this whole idea of health economics. If you can improve technology to the point that patients' lives are better and it's cost effective, then it becomes something that we should be adopting.
No, that makes perfect sense.
Yeah.
So you and I, we grew up in a time where it was science fiction. We watched Star Trek where they had the little tricorder that they ran on somebody and said, "Here's what's wrong with you." How far away are we from something like that? Do you think that we'll have a universal panel that will tell us a lot about what's going on?
I don't know what the capabilities of that device are going to be. Is it getting things that are floating out of the air? Is it doing advanced AI informed signal processing of all the conditions that are being? You can see parts of that are happening already. In some ways.
Yeah.
But I think we are complex organisms. We are very complex.
Yeah.
And figuring out how we actually work, you got to understand the basic biology and the physics of what's going on before you can build really meaningful things, I believe.
Yeah.
And you know this in drug discovery.
Yeah, absolutely.
It's all based on this. The physical chemical properties of the new molecular entities that you build have certain functions that they can actually alter that take place in cells. It's just that's the way it works.
That's exactly right. Yeah, I tell people all the time, what I do gets reduced down to, in biology, a fundamental principle of biology is structure drives function.
Yes.
And what we do is change, and in fact, what mutations do is change the structure of proteins, right? So you change the structure, you change the function. So drugs, that's what they do. They change the structure, they change the function. And that's what you reduce it down to. But that's exactly right. The physical chemical properties of that molecule that we're introducing actually induces a change in the structure of the protein, which changes its function. And even more complex biological drugs, which is kind of the newer, fancier drugs that we're developing that aren't the old school chemicals that we used to make, that we still make. I still do a lot of that, but they're still doing the same thing because that's biology. That's how biology works is we turn things on and off by proteins interact. As you mentioned earlier, proteins are the workhorses of our body. When you look in the mirror, what you're seeing is a bunch of proteins. When you go exercise, it's a bunch of proteins that are making that possible. And our drugs, what they do is change the function of those proteins. So either we turn stuff on that should be on that's turned off, or we turn stuff off. Most of the time, we turn stuff off that gets inappropriately turned on. So I guess at the end of the day, our diagnostics are kind of centered around trying to determine whether things are on or off that should be shouldn't be that way, right? And
it's.
What if you could get rewarded just for being healthier? Meet Galvin, the company using the latest blockchain technology to reward you for taking care of yourself. They help with your mind, body, and soul like a personalized health assistant to track your progress and help you feel better. Galvin's G Plus app rewards you for exercise and assesses your mental health in under a minute using AI and over 2,500 voice biomarkers. If you're feeling off, G Plus gives you personalized power-ups, science-backed techniques to help you feel better fast. And here's the best part. When you prioritize your wellness, you earn crypto. Ready to level up your health? Use promo code GOODMED20 at checkout for 20% off. Download G Plus today at the Apple app or Google Play Store and start your wellness journey with Galvin.
Really interesting. And in pregnancy, I thought this was a fascinating aspect of studying early pregnancy. Can you predict the changes that actually lead to early delivery? Preterm birth occurs in the U.S. 10.4% of babies are born too early.
Yeah.
And they can have both immediate and long-term complications and higher mortality rates too. So if you're able to actually identify those pregnancies early and intervene early, you can get there. And the diagnostic test that could identify them has led to prospective studies that show that you can benefit the babies. And at the end of the day, the well-being of babies is important. David Eddy taught one principle that I've never forgotten, and I should have articulated this at the beginning, and that is that oftentimes we focus on intermediate outcomes. I'll give you a simple example: blood pressure.
Yeah,
we want to measure blood pressure. We want blood pressure being in control. Blood pressure is just an intermediate outcome. It's a measurement you do.
Yeah.
What are you really trying to improve? You want to decrease the risk of heart attacks, stroke. And kidney failure.
Yeah, that's exactly right.
That's what it is.
Yeah.
So the long-term outcomes, if you build technologies that improve the long-term outcomes of patients, and for cancers, it's recurrent. You want the patient to have long-term progression-free survival. Those are the kinds of things you look at in the studies that you build. In your area, you're going to look at being able to increase the mental function of an individual for a longer period of time in his or her life.
Exactly.
That's what you're looking at.
Yep.
Those are the long-term outcomes that your drug has the potential to ameliorate.
Yep, that's exactly right. No, that's a great way to put it, and that's where you can make the biggest impact is looking at these indicators that are going to tell you what's going on long term. And I guess that leads to the next thing I wanted to ask you about. So there's a big movement going on right now about longevity.
Yeah.
So we're starting to understand, and I think all of us realize that some people live a long time and some people don't live as long, right?
Yes, that's right.
And there's populations of people that live longer than others. And we've been studying this for a long time, but it's becoming a bigger deal. And part of the reason why it's becoming a bigger deal is we actually are having less children than we used to, and our population is going to start declining. And we're already there, right? And so just from a pure civilization and economic standpoint, we've got to keep people healthier longer. And so thinking about long-term and longevity, this has become quite a hot topic right now. So from the diagnostic side of things, there's a lot of work being done right now on trying to figure out these molecular clocks that how fast are we aging, and so tell me what your thoughts are about how easy is it going to be to adopt these things, and what's going to drive the uptake of this, and what impact do you see it having on people changing their lifestyle, changing how they approach thinking about keeping themselves healthy. In fact, I think one of the big buzzwords today is "Make America Healthy Again." Right? Is that we're trying to there's a bigger focus, I think, on not just waiting for people to get sick and trying to come up with treatments, but how do we keep people healthier longer?
You want people to have longer, more productive lives.
That's right.
That's what you really want to have. So in some ways, I'll just disclose this. I'm also working with a company that does epigenomics.
Uh-huh.
And in fact, the first person to actually use methylation to find cancer in blood is the founder of this company, and it comes out of the UCLA.
Uh-huh.
And one of the things we talk about is you can actually give an age, a physiologic age for an organ by using the methylome.
Isn't that interesting? Yeah, it's amazing.
But it makes sense. I mean, we know that all these things are connected in some sense.
Yep.
And the question is, what are the signals? Can we read them, and can we use them?
Yeah.
That's the way I think about diagnostically. And even for longevity, what signals are there, and how can you follow that, and what will you actually be able to do with that will actually make lives better, make people live more productive lives, and be able to have a longer lifespan. There are a lot of questions that are unanswered still, but the fact is we have powerful tools, we have a lot of data. We just have to feed it to the AIs and clear thinking scientists to come up with ways to do things better. That's what we have to do.
I completely agree, and it's the data that's coming out around this, like you said. It's pretty compelling. Like you can tell the difference to say, I might be 56 years old, but my liver is 70.
Precisely. That's the kind of information that's possible.
Exactly.
But how would a consumer use that?
Exactly.
And what will doctors say about it? Those are the big debates that you have when you're actually talking about these kinds of tests in these companies. What will the doctor advise the patient to do?
Yeah.
What would, if you had a very knowledgeable doctor armed with the best AI interpretations, what would you tell the patient to do differently? We don't know the answers yet.
Exactly.
And here's the unfortunate thing that is both a protection and a hindrance for progress. The protection of patients is you don't want to do something that's going to harm somebody.
Yep.
You never want to do that. But not looking at these things and not trying them out it means that you're going to miss opportunities. And so I think what it means is we have to find ways to fund research to look at these big questions.
Yep.
That we've got to find ways to do it.
Yep.
And some of this is at the basic research level. Other is at the application level where you can actually do something with it. Most of the funding right now in biopharma is at the application level, not so much the basic research. But we need to answer these questions, and I don't think we're addressing them strongly enough yet.
Well, I couldn't agree more, and I think that one of the challenges, especially on this longevity question right now, it's fascinating to watch this happen because a lot of it's being driven by very high net worth people that are investing in things to benefit themselves directly, to say, I want to live forever. And one of the things that I just can't stand is health disparities. I can't stand it. I can't stand that we have technologies that are only available to people with money. That really bothers me, right? And I think that's the problem with the direction we're headed right now, where we're cutting basic research, which could foundationally help everybody, and really it's being picked up by people that are on the high net worth side of things that are really doing it to benefit themselves more than
and that's a problem. It's an economic problem all over the world. And we saw this with BRCA testing in the early days. We were criticized severely for not making the testing available to third world countries. Well, guess what? Their health systems couldn't even use the information if you gave it to them.
Exactly.
You gave it to them for free. They couldn't do anything with it. They didn't have the doctors to follow up, the screening technologies to do anything about it. So it didn't make any sense at all. There are huge disparities around the globe. However, I think one of the things that inventors of technology need to think about is, what is the migratory path to get the cost of goods down to the point that you can democratize these technologies to everybody? And that's one of the things that you try and do. If you can get the cost down, then more people are going to be benefited, and it will reach into lower strata of the socioeconomic scale and help other people.
Yeah, no, I think that's exactly right. And part of that obviously is as we adopt these things, we get better technology, which typically does drive costs down.
Does.
Right?
It does help. It does help.
So today, running the BRCA1 test versus what it was, I mean, it's night and day different.
It is night and day. Yeah. At the time, when we were running Myriad, we were the highest quality, most economical sequencing laboratory in the world. And nobody could sequence as fast as we could.
Yeah.
And now you kind of laugh at the goalposts that we hit along the way. Well, I remember in 2006, I said, how long does it take us to sequence a gigabase of information? And he actually it was a megabase then. That's what it was. It was a megabase.
Megabase, yeah.
Okay. How long does it take to do a megabase? It took us a year and a half to do our first megabase of sequencing at Myriad.
Yeah, incredible. And one of the best places on the planet.
And he said we do it in a couple of hours in the morning now.
Yeah, exactly.
And that was in 2006. Think how far we've come since then. It's 20 years later now.
Yeah, so in a few minutes.
With next gen sequencing, things have just continued to explode.
Yeah, no, it's a remarkable time to be involved in what we do because we've seen just a revolution in technology and how that's changed what we can do. I know there's tests that are coming out that can not only predict risk but recurrence of disease and in the cancer world. We have circulating tumor DNA and modified DNA and all of these things. Just amazing. It's
a lot of work on liquid biopsy.
Yeah, liquid biopsy. That's a term that a few years ago nobody knew what that meant, and
right.
it's kind of in the standard lexicon in the medical world now. That instead of going in and biopsying the tumor, which is terribly invasive and sometimes not even practical, right? I mean, it's challenging. We can actually get a sense of what's going on just by drawing the blood.
Well,
right?
and in fact, you could make the argument because cancer is such a dynamic disease and the changes that occur from the primary tumor when it's first discovered, you're probably better off doing the blood because you're getting more representative of what's happening outside the primary site.
Exactly.
Because it may have changed.
Yeah.
And in fact, there are lots of tumors where that's been shown to be the case.
No, it's just the ability to start looking at these things. So it's the best of times and some ways the worst of times. We're dealing with a lot of uncertainty about the future of research and the future of who's going to fund these things. And you and I have both been entrepreneurs and started companies. And I mean, to be honest, it's the hardest time that I've been involved in.
It's not easy.
It's not easy right now, even though we have all of this opportunity and all this information. And but we wouldn't be who we are, and if we weren't the eternal optimists as well. So I think that's part of what keeps us going. Is we hope for the future that things are going to be better.
Yeah, I think optimism is a requirement to be an entrepreneur. You have to be optimistic, and you have to have boundless energy.
Yep.
And you can't be discouraged.
Yep.
And when you're on the track of something that's important. You don't let anything get in the way.
Yeah,
that's what you end up doing.
Yeah, no, and it's a passion project, right? You have to develop that you live and kind of breathe this stuff, and that's what I love about you is we're both getting older, but I still see the fire inside of you. You still love this stuff, right? Well, I mean, it gets you up every morning and gets you excited.
I think about it constantly.
Yeah,
there are ways that we could do better. And one of the beliefs I've always had, you mentioned science fiction. I believe that we're seeing yesterday's science fiction become today's reality, and tomorrow will be the same way.
Yeah,
and there's always a better future ahead of us, but it's up to somebody to create it.
That's right,
and that's what we have to do.
Yep. No, that's exactly right. Well, I think that's a great place to end. Actually, is to say that that's why we're here, and that's why I love having these discussions, meeting people like yourself, and being able to talk about the fact that somebody has to do this, and meeting the people that are actually doing it out there on the front lines. And I mean, what you've done and the lives that you've changed, it's just remarkable. But what's exciting is how much more there is to do.
Well,
right?
The one other thing that I think we need to state too is this is not done by a single person.
No,
it's
absolutely.
done by teams of people.
Yep.
The number of dedicated people that it takes to push forward these initiatives is huge.
Yep.
And you couldn't do it without dedicated, hardworking, smart, talented people to help build these things. It just doesn't self-assemble.
No, not at all.
It takes an entire team and people both inside the company and outside the company that catch the vision and want to be part of it and help it move forward. That's what you have to do.
Yeah, and I think that's exactly right. And I think for all the bad rep and that sometimes we get in the pharmaceutical industry and just the medical profession in general, I don't know a better group of people, more dedicated and focused, just collectively, than the people that I interact with on a daily basis. It's amazing. Like people are not doing what they're doing to make money or to try to, it is because they love what they do. And that's what keeps me going, right? Is that I have a bunch of young people downstairs that are just, I mean, we had an intern that just reported and was literally crying, saying how much this experience changed her life. So it's fun. I love what we do, and making good medicines is what we're all about. So I appreciate you coming in today and talking about something that we don't talk a lot about. The diagnostic part of medicine is super important. It might be more than half the battle is figuring out what's wrong with people, and you've made a big impact in people's lives, and really appreciate everything that you've done.
Well, the best is yet to come.
The best is yet to come. That's right. Thank you so much,
Thank
Greg.
you.
Yeah. That's it for today's episode of Good Medicine. Thanks for joining us as we explore the science and stories behind health, medicine, and innovation. If you enjoyed this conversation, please subscribe to our YouTube channel. That's a great zero-cost way to support us. In addition, please click, follow, and leave a review for the podcast on Apple and Spotify. If you have questions or comments for me, please put them in the comments section on YouTube or reach out on all social media platforms at Good Medicine. Links are in the description. The best way to support the podcast is to check out the sponsors of the show. You can do this by looking at the links in the show notes. That's it for today. I'm Dr. David Beers, and we'll see you next time.